want to thank the AIS for the opportunity to speak today. Let me see if I can move my slides. All right. Well, one of the common theme today was the rise of GLP, and yes, as a result, the world is changing because of that. The treatment protocols are no longer about the very isolated aesthetic correction. It's now about treating patients individually based on their age and biology, and it's no longer the cookie-cutter treatments, standardized treatments that we've been seeing in the industry for quite some time. We're in the world now where we need more predictable consultation, data-driven treatments, and then risks that are well understood before even procedures can even begin. And that world is not near distant future. It's close enough for us, and it's already taking shape, and the AI is in the forefront of it. Some of the technologies where AI is being used in aesthetics already is happening in diagnostic and imaging side of things. In treatment solutions, for example, tools like Vectra and Modiface by L'Oreal has already been used in that area. There's precision robotics being used for that. And then in personalized skincare, a lot of apps are coming up as well, too, and people are using that to learn more about their journey before treatment, after treatment. But there's next opportunity, and that next opportunity is in the product development space. And given my background in neurotoxins, I'm going to touch upon that today because neurotoxins are still, in my mind, a foundation for the aesthetic patient journey. Back in my Oregon days, we used to actually refer Botox as the gateway product, which I think is still the case today. Earlier today, I was talking to a dear friend, David Mozzaretti, about all this influx of neurotoxin products. There's about 7 approved in US, EU, roughly speaking, and Relabot it's got approved in EU, not in US yet. But nevertheless, they're all targeting around the same audience as well. So where is innovation in this space? What we need truly in neurotoxin, particularly a true step change differentiation. Most fallen programs are what I call incremental improvements, not paradigm shifts. Whether they're defensive innovation, you will see some liquid formulation will come in, or a long duration of a toxin, for example, which may reduce visit frequency or whatnot. One of my claim to fame was building Bonti. We were developing fast onset neurotoxin, which could be disruptive and have a short duration, will have its own purpose, and kind of looking forward to that approval one day, and hopefully some of these, the real innovators, the practitioners will figure out a way how to even use that in new indications. Just like how Botox grew, we're expecting the fast onset duration neurotoxin will also grow with the same sort of journey. And there's some niche expansion happening as well, going after new areas of the face, or going on the neck area, or other areas where there are new applications of neurotoxin can be understood. But the comparative battles are not just about efficacy parity. It's about, in the end, patient experience, and for companies, for strategics, how do they expand their portfolio breadth? My journey started when I was at Regeneron as a scientist, and that's where I got first exposure into protein engineering and novel antibody and bispecifics and so on and so forth. And those were designed to go after certain challenges, go after certain characteristics, right? So hence, protein engineering has been around for quite some time, where some of these companies will use their drug discovery groups and using AI in some cases as well to go find new applications and make improvements to a biologic, how it delivers certain targets and so on and so forth. On the right, you can see there's a protein design, and just by tweaking a few different things here and there, you can improve efficacy. You can reduce toxicity. Just by pH-dependent cycling, you can even improve half-life or injection frequency or dosing toxicity. Some of the elements can apply to what we do in aesthetics as well, too. As a quick refresher on the course, the protein, and I'm sure some of you guys this might be new to you. It is a complex structure, but there's a lot of interesting elements to it as well. And if you think about learning languages, starting from letters first to forming words, to forming sentences, or even forming paragraphs, Similarly, the proteins around the same concept as well start with the amino acids. I'm sure you guys know that from your biochem class from college days, or the protein sequences. And then they then turn into some complex single-domain 3D structures, and then on multi-domain structure as well too, and which is where, as you see, some of these biotech products are now designed as such as well. Some are bispecific, some are trispecific, going after different targets. And similarly, if you take this concept into neurotoxin, you start with one sequence, you let the protein engineering, protein language do its science, and then it'll turn into an output, which is a neurotoxin with the desired characteristics and effects. And protein languages can play an important role in improving affinity and functioning, which may lead to lower toxicity or lower immunogenicity or even higher potency as well in some cases. As a refresher on the neurotoxins, some of you guys may know there's several serotypes of botulinum neurotoxins. A is obviously everyone knows from Botox and other similar products that came after that. It targets SNAP25. It's part of SNARE complex, which has 3 key proteins, SNAP25, which is where A is, VAMP, which is BONT-B was, or a product called MyoBlock, which came out many, many years ago. It wasn't quite successful in the aesthetics, as it was touted as very painful injections, and a lot of physicians didn't use it because A was offering similar attributes, so it didn't make sense for it to be pursued further. And then there's type E, which we developed at Bonti, very similar concept as A, same MOA, but has a fast onset and short duration as well. And on the bottom, you can see there's sequences that they all have own unique, basically, attributes to it, which allows them to behave differently, and hence will then react differently as we use in different applications. One of the approaches that have been taken these days is recombinant. There was a time where recombinant was seen as not provide same potency, same attribute as a wild-type strain, but some of that has changed. A lot of research has gone into space. There's actually a product coming out of China, just got approved in China, I think, purely recombinant, and is claiming to have same attributes, same profile as a wild-type Bonte, like Botox product. But what recombinant also does, it improves from your production perspective, may also reduce some toxicity, and will provide better safety profile as well too. You can also file IP around it as well too, around unique uses, unique application of it, and more importantly, you can also then dose as you dose other biologic products by mass, not by unit, so you don't have that debate anymore about what unit means in this case. Ipsen came out with a product. In fact, not too far from here, Dr. Min Dong's lab first developed this hybrid chimera, which combined Bont-A and Bont-B. And you can see there are other combinations as well too, but the unique thing about that approach, what Dr. Dong's lab did, was that it basically took the strengths of a Bont-B because it has a very high affinity, enhances the receptor affinity and internalization of the neurons, which helps with the potency as well too, and also allows better diffusion of the program as well too. And then when you combine with the light chain of the Bont-A, it improves the pharmacological properties of the overall Bont products. So what should be the attributes of a neurotoxin, ideal neurotoxin? I've been asking them this question to my fellow drug developers, to physicians, and even some patients as well too. And then there are 4 prime factors that I sort of kind of came up with based on this conversation. One is precision controllability. Predicting dose response is important, localized effect as well too. And then obviously in some cases from a physician perspective, improved stability, storage, and transport, where you can keep it at room temperature, for example, that obviously helps. And then high safety margins are another element that came out of that. Predicting your onset, predicting your duration effect is very important as well. Ideally, if you can have something that comes within 24 hours, it's great. And then the other key thing that came up was the longer peak efficacy, something that lasts 4 weeks, but then have a quick drop-off. And I'll show you a slide that will explain that in more details. And then there's also a patient aspect as well too, the risk tolerability element, and maintenance versus event-driven. These are all things that factor into what the real ideal neurotoxin attribute should look like. And some of them will come in with the refinement of existing products, but as well as engineering and combining a couple of the serotypes that I mentioned early on. This slide kind of shows you, and since Connor's going to be speaking after me, so I'm going to give a little plug-in for him on that. On the left is the BONT data that was presented by Allergan, where you see the fast onset. Within 8 hours, you see the onset coming in. And on the right side, you have the BONT, which has longer duration. I imagine if you have something that comes in quick and then lasts for a longer period of time, where the peak effect in this case, which is lasting a few weeks, can be extended further down and then have a quick drop-off. This would be an ideal product for both patients and the practitioner perspective. So in summary, where AI can be used for idle neurotoxin from data collection. We know all the sequences. We know the historical data that can be input. AI can help streamline some of that. The sequence can also be identify where substitution can be made to make it a bit more core to the original snare complex properties. Protein engineering can help clean up in terms of removal of misfolded proteins and unstable conformation, which are likely to aggregate. Functional property prediction using machine learning can also help in terms of how the program or this molecule will behave biologically, where it should bind, where diffusion should look like, and then onset duration can be predicted with that model. And then with multi-objective optimization, you can have a balance as well. Should I increase it from the toxicity perspective, or if you're keeping diffusion in the same place by getting the potency high as well. And last but not least, you can also then use it for more clinical production as well. It's been used in clinical trials as well, as a lot of people know that already in terms of patient selection and overall optimization of clinical design. But ultimately, where the future is in essence, right? And this is where, as opposed to having a me-too products or fast follower products, we need some new innovations, and I think AI is in the forefront of that, where we can bring precision, safety, control, and more personalized treatments will eventually lead to an ideal scenario that is not just in neurotoxin, but also in other similar products as well, too. If you have any questions, I'll be out there. Love to talk to you more about this, but thank you so much, and I'll let Connor talk more about the neurotoxins. Thanks.